Welcome to my world, the world of science. Knowledge is power, health is wealth. We know there are so many mysteries in the world, but so many of them have been unraveled by science. Medical laboratory science has played a great role too. And one of the mystery of the world that has been unraveled by medical lab science is food poisoning. Ordinarily, you will think when you talk of poisoning, somebody has poisoned or has used something very untoward, very unethical, very wicked thing to harm the other person. But in this wise, what is harming, what is producing the poison is invincible, and it's only the aid of the laboratory, the medical laboratory scientists, that can really prove the cause. And this is what I want to expose. This is what I want to demystify. This is why you have to watch this video to the end. What is that which is somehow added that now cause that food poisoning problem? The symptoms you see, the diseases you see after eating certain food that has food poisoning. So let's go on to the slide. Food poisoning demystified. Yes, in this wise, it is toxin that is produced.
Definition. According to WHO, foodborne illness are usually infectious or toxic in nature and caused by bacteria, viruses, parasites. The normal understanding is there is that poison produced, and the poison in these situations are called toxins because bacteria produce toxins. When you talk about chemicals, you now talk about poison. So natural toxin in plants and animals such as mushroom and shellfish or harmful chemicals, that's where such agents such as insecticides and heavy metals, these are the ones that really comes in as poison entering the body through contaminated food. Chemicals contamination leads to acute poisoning. The symptom of foodborne illness develop within a period of several hours to 2 days after eating contaminated food, and usually includes the symptoms the listed below. Food poisoning means specifically illness caused by toxins. The bacteria produce that toxin, like I had earlier explained. Toxins are produced mostly by gram-positive organisms and are called exotoxins that act locally and can spread far, whereas most of the gram-negative organisms have membrane-bound toxins called endotoxins that are released into the environment upon disruption of the intact bacteria, or you can call it cell lysis. Endotoxins are lipopolysaccharide covering found in the outer membrane of various gram-negative bacteria. This is the gram-negative. It's always pinkish when stained. We know the principle. And this is the gram-positive organisms, bacteria. It's always purple, blue. Food poisoning. I'm going to try to explain these 7 listed food poisoning type: the botulism, the bacteria or infectious diarrhea, E. coli, enteritis, fish poisoning, listeriosis, salmonella, and try to talk about staph too. But mainly I shall take just about 5 of them. This is botulism. When you are now talking of the Clostridium organism, the Clostridium have there are 4 spore forming. These are their spore. You see the rod shape, but it forms spores. These spores help them to withstand harsh conditions, and so in their vegetative state they produce this poison, or we call it toxin. And the toxin they produce affect the muscles. So you find it when it's very difficult to breathe, slow speech, these are all muscular movements, trouble swallowing, the eyelid, sleepy eyelid, blurry vision, and weak muscles. The disease caused by Clostridium botulinum are caused entirely by the neurotoxin. The neurotoxin, which we just mentioned as exotoxin, there are 7 antigenic distinct toxins designated A, B, C, D to G. They are resistant to gastric acidity, and when they enter into the bloodstream, they are the most deadly of bacterial toxins. Death usually occurs from respiratory failure, because when the muscles cannot really contract like that, then the person cannot breathe, more or less, and that means death is so near. Oxygen is very important to life. The toxin acts by blocking acetylcholine release of production at the synapse, and makes it difficult for the movement of the muscle. Flaccid seed paralysis results. Infectious diarrhea is the next one, which is very obvious, has to do with bacteria or infectious organisms. And this is a table that really shows the ones that are causing inflammatory reaction and non-inflammatory reaction. Both of them will cause diarrhea. You have it, the inflammatory one, or toxin producing pathogen. These produce toxins and mucosal damage and tissue destruction. So this is where you find organisms like Salmonella, Shigella, Campylobacter, hemorrhagic E. coli, invasive E. coli, and Shiga toxin producing E. coli, Clostridium difficile, and Yersinia entero. The parasite is Entamoeba histolytica. So these really are the bacteria that produce toxin, and the parasites in this area that really cause mucosal damage. There is the Entamoeba histolytica. You find it around the large intestine. These other types are more or less not as invasive as this, but the mode of the mechanism of action is they increase the secretion. We have the rotavirus increasing the diarrhea, norovirus, and majority of these are found in children. They cause problems mainly in children. Even the E. coli too is more common in children, to vibrio cholera in adults too. The severity with the non-inflammatory is mild, but with the inflammatory is typically severe. Bloody diarrhea is more or less called dysentery. This is Campylobacter shape, jejuni. That's one of the bacteria producing toxin, Campylobacter jejuni. And the mechanisms of pathogenesis of Campylobacter jejuni comprises 4 main stages: adhesion to intestinal cells, colonization of the digestive tract, invasion of targeted cells, and toxin production, thereby giving rise to increased level of cyclic adenylate. The adenyl cyclase is what stimulates the transport of sodium. So you find more water, since this will continue the production, so more water will now be flushed to dilute the sodium, and that's where the experience of diarrhea. Once sodium is being transported, you find more water really being for water movement to dilute the sodium, and that's what you call diarrhea. The fish-borne poisoning is usually common with this Cichlidothera fish, and it is not bacteria, it's like a fungi, mold poisoning, like a mushroom mold poisoning that really cause serious problem. 90% of cases from Florida. Most common species are carnivorous reef fish, barracuda, and the popular barracuda, I'm sure. So this is also a very common fish poisoning. And then when you talk about canned foods, canned fish and what have you, there are other organisms like Listeria monocytogenesis and others that produce these toxins, and they are there to really cause a serious problem by the time you eat this kind, open the can and eat it. So we have listeriosis, Listeria monocytogenesis. Cytogenesis is a serious one too, very serious problem. It causes septicemia. Once something can cause septicemia, it could cause meningitis, and it could cause kidbirths, pregnant women, and as a result of unsanitary commercial processes of dairy, like I said. So this is the shape of when you can stain it, unlike the other shape where it is full of flagella, peritricious flagella surrounding it. Like Listeria monocytogenesis is generally transmitted when food is harvested, processed, prepared, packed, transported, or stored in environment contaminated with it. So you could also eliminate it in fish poisoning. The weight mechanism is more of exotoxins, such as listeriolysin, very close to the streptolysin G. So the mechanism is almost the same, happening if you get in Listeria infection and sepsis. Now, salmonellosis. Salmonella are gram-negative multivacilli and fall into two species, the enterical and the fungorin. The foodborne pathogen that really cause human disease range from mild gastroenteritis to severe, caused by the enterical and the typhimurium. So the other way of classifying them is to call them the typhoidal salva, which is the Salmonella typhi, paratyphi, A, B, C, are really causing problem in human and enteric fever. But the non-Salmonella salvas are the Salmonella typhimurium and enteritidis have a broad range of hosts which they affect poultry and other species. This is the shape of the organisms. You can see the flagellum. The plasmid is part of the things it uses to resist, and every living cell must have a DNA to really perform the genetic function of bringing out the production of proteins that is also essential to life. All these are proteinous the cell wall to be the amplifications of so many cells when you have macromolecules joining together to now form that big. But the message is gotten from the DNA. And and this cytoplasm. So it's well labeled. And, foodborne salmonella toxic infections are caused by non-typhoidal salmonella cerevisiae, typhimurium, and enteritis. And so the area that concerns us is also where you now think of the diarrhea and vomiting that comes out from these two species, typhimurium and enteritis. So the laboratory diagnosis is as simple as we're trying to perform a neutralization test of the toxins, antitoxin, and then you'll be using the food that is containing the toxin. And there is also the agglutination test too. You can also do ELISA. It's almost the same principle like serological test. Then the common microscopic culture and sensitivity on the patients to do more. And the prevention is just to make sure that the food is well cooked, and food handlers are people who maintain high hygiene, very good hygiene and sanitation, as well as food handlers should also be tested for common like salmonella organisms that they could be carriers. So we don't want that happening anymore. But since we are available, it could also be given to people, especially in developing countries.